Professor Magistretti, Professor Barros, Professor Beyer, Professor Linne. I see that you're all here. Welcome, Felipe. So,
hey, Corrado, how are you doing?
Good, good. Thank you very much. So, thanks to the members of the panel for being available for this friendly discussion. So, the title that has been given to this session is the future of glia research. So if I can quickly introduce myself, I'm Corrado Cali. I'm an assistant professor at the University of Turin and also a Magistretti scholar. So today I will moderate quickly this session, and I hope to do a good job. I have an incredible panel in front of me. So I think one good way to start this conversation, because we're somehow talking about astrocytes, which is the glue that is sticking us together, just to use a term that reminds us of glia. A good way to kickstart would be maybe to ask a panel to discuss a little bit their perspective and their take about how calcium and metabolism are somehow linked together, because I think these are two paths that somehow has been running in parallel, but never really met. There's been some not a big consensus about how the language of calcium, which I think most of glia researchers think that it's the way astrocytes are communicating together or with neurons, and metabolism, which is a way astrocytes are providing energy to neurons. There's not been much consensus about these two are going together, and some recent papers have been shedding lights onto the molecular mechanism through which somehow intracellular calcium can be related to metabolism and to ANLS in the brain. So, what's your take on intracellular calcium, on calcium language, and metabolism and the ANLS? So, does some of you wants to take the ball, or should I pick up?
I can just mention a paper which was published a few years ago by the group Maiken, Nedergaard, and I think only one other author. I think there were only two. And in cerebral cortex, where she stimulated the locus ceruleus and showed massive activation of astrocytes in terms of calcium transients and waves. I forgot somehow the details of the paper, but that paper reminded me that calcium, I mean, glycogenolysis can also be triggered by calcium, and in fact we and others had shown it. So that if you take that work, but the Maiken didn't explore any further between, it was just calcium waves triggered by stimulation of the locus, which makes a lot of sense, noradrenaline mediating this effect. And as far as calcium is concerned, it's mostly an alpha receptor mediated, and calcium can trigger glycogenolysis. So I thought that was an interesting paper, which I think connects very nicely with, I think Felipe mentioned the work of Bruno and Zand and others, recent one, again, activation of the locus and attention and arousal, which goes again back to the very old proposal that I made in my thesis, that the noradrenergic system is there to kind of prepare the cortex metabolically to incoming specific activations, and this can be Um, and I think calcium can play a role on this. So this is the first point. The second point is, again, a few years ago, I think in 2002 or 3, I forgot, we published a paper, and the first author is Jan Bernardinelli in PNAS, and showing what we call the glucose and the sodium and glucose waves. So that was an in vitro study where we triggered, we electrically activated or even mechanically activated in astrocyte, that was with Jean-Yves Chaton, by the way. We triggered a calcium wave, and the reasoning was, okay, this calcium wave should trigger release of glutamate, because that had been shown by Volterra and others. So Hayden, Parpura, you name it. And so then we said, well, okay, this glutamate that is released by astrocyte will be somehow taken up again by astrocytes, and this will trigger sodium transients because of the uptake site involves sodium. And since we could visualize sodium, we said maybe we see a sodium wave following a calcium wave. And then we said, well, if this is true, then it should be like a local ANLS triggered, an astrocyte. It's not AN, but it's AALS, astrocyte lactate shuttle, whereby actually not lactate, we don't know, but anyway, that glucose could also be taken up because if sodium increases. And we saw, using NBDG, a sodium wave, metabolic wave. So the connection that we found was calcium wave triggers a glutamate release, and then a sodium wave, which then triggers a metabolic wave. And just for the anecdote, we submitted this to Science. Obviously, it wasn't even sent out for review. We sent it to PNAS, came out. The week after it came out, it was in Science as the editor's choice. Yeah. So you could have had it in Science if you wanted. So this is what I can contribute to this this aspect of calcium and metabolism and astrocytes.
Thanks, Pierre. I was curious. Maybe I would direct the question directly to Felipe. So because you've been developing the laconic, the pyronic. So you have a very visual way, a very visual approach on studying the dynamics of lactate. So have you ever, to your experience, done any experiments where you try to visualize the flux or the concentration of lactate and then relate this to calcium waves?
Yeah, actually, we started years ago looking at calcium, of course. Calcium is everywhere in neuroscience, so it must be calcium, right? And then over the years, we are more and more confused. Let me explain. Of course, if you induce a very strong calcium signal, say with ATP, something like that, of course you get impact on metabolism, but it is usually negative impact. So you get mitochondrial inhibition. So lots of calcium goes into mitochondria and the cell stops using oxygen, and then you see a huge activation of glycolysis. So when we first saw that, we thought, oh, this is very interesting. But then reading the literature on calcium and how nuanced it is in terms of what is the physiological calcium signal, we don't know. I think calcium is doing different things at different concentration on different time scales. So maybe those very small calcium transients Marja-Leena would see in an astrocyte process is a different story than the whole the huge calcium signal you see when you activate the locus coeruleus. And we, of course, we try to integrate this in one story, and I think it's time to really we need to be more detailed in terms of what calcium we're talking about. Of course, you know this very well. The origin of calcium, what people, electrophysiologists have known for a while, we should be incorporating this into metabolism. But my feeling right now is that you have this calcium world in parallel to the non-calcium world in terms of regulation of metabolism. So every time we tried ammonium, potassium, even nitric oxide, calcium doesn't move. So if you activate an astrocyte with potassium, you have strong activations of glycolysis, a factor of 4, and calcium is flat. Of course, it could always be a microdomain in a micro corner that is invisible to us. But my gut feeling is that there are parallel ways in which calcium is doing things, and we will probably be able to integrate this into metabolism when physiologists, calcium physiologists, are able to tell us what is a physiological calcium signal, right? And right now, you know, this is a controversial field. Some people say that Glio transmission mediated by calcium doesn't exist, the others do. I think, well, there are recent papers in Neurons by 2 or 3 groups which converge into concluding that mitochondrial calcium was a figment of somebody's imagination. So 20 years ago, mitochondrial calcium was the big thing. So mitochondrial calcium would rise, and then the dehydrogenases would be activated, and all of that is really crumbling, particularly after the MCU knockout animal was produced, and people started doing the experiments, and they found that the animal doesn't have a phenotype, not a neural phenotype. And then going to do the experiments, apparently mitochondrial calcium is not very relevant, at least under physiological conditions. And then you have this work by Georgina Sadrustegui and Norbert Gellerich, that they have shown that extra mitochondrial, say cytosolic calcium, would be important. But it's very difficult to reconcile the time course of the signals with what happens in metabolism. I will give you one example. Sorry, I'm getting, this is too long, but I will finish with one example. We stimulated neurons with a very decent protocol, which is called the short theta burst. With that protocol, we get a 3-fold stimulation of glycolysis, a 3-fold stimulation of mitochondrial pyruvate consumption. And the time course of those stimulations is slow, like metabolic stuff, right? Seconds. So mitochondrial pyruvate consumption goes up with a half time of 6 seconds. Glycolysis is activated with a time course with half time of 15 seconds. Then you look at calcium, cytosolic calcium is up immediately, milliseconds, and then down. When all the metabolic things are happening, calcium is down to normal. And now you look at mitochondrial calcium and it doesn't move under that particular protocol. So if it is calcium, it's doing something very clever. It's not like metabolism is following the calcium signaling. Calcium might be priming, might be starting something that is really regulated by something else, maybe sodium, because sodium follows the right kinetics, as Pierre was saying. But it's super complex and difficult to tackle. It's like, in this moment, it's like 2 worlds that they're not conversing to each other.
Yeah, this is a very good point, and in a way, what immediately when you were talking about this last experience, what was coming back to my mind was probably the last Volterra's paper in Science, where he analyzed in 3D different patterns of calcium on different microdomains, and this is something you have said already. So it really depends on where you're looking at. Because the compartmentalization on astrocytes is extremely important, and we still have no clear picture of where the different compartments are contributing to the calcium language, and how. You have mentioned Glio Transmission, of course, and Glio Transmission runs at a different time pace compared to metabolism, of course, because it has to go at the same speed as neurotransmission, or at least something similar, and metabolism is much slower. So I think we can hypothesize probably what would make sense is that to some extent calcium related to glio transmission then feedbacks back to astrocytes through glutamate, and then sodium would make the rest. Why not? It's as you said, it's pretty complex, but might be an idea to investigate. So,
you know, sorry, complementing that.
Go ahead.
You know that there are many mysteries regarding this is maybe relevant for Marja-Leena's models. If you look at the sodium pump, in most models, the sodium pump is regulated by sodium, right? That would be the regulatory. Mm-hmm. But it's not true. Because in neurons, if you model the sodium pump, you will see that the increase that is actually observed in rate, typically say 300% from 10 to 12 millimolar sodium, that is totally incompatible with the regulation by sodium. So even though we talk about the sodium pump, we don't know what is really regulating the sodium pump, and it's not sodium. And then, so something more complex is happening, and maybe calcium is part of the answer. Because if you do the experiment in the presence of EGTA-IM, in which you inactivate the sodium entry neurons, you don't get the activation of the sodium pump. So somehow calcium is doing very clever things which are related to the concentration, and it's like a priming, like a kick-in effect, and eventually I know your models are very complex as they are, but eventually you may like to include that very delicate kinetic analysis into the model, because otherwise the model will not work very well.
I mean, I think what you're saying, and I fully agree, is maybe we don't know enough about calcium dynamics in space and time. So to link it to metabolism, it's tricky. So we can make speculations, interpretations, but we probably have to know more about calcium regulation in space and time in astrocytes.
Which would lead me to shoot the ball to Marja-Leena at this point, I think, because I mean, many of the dynamics that we're looking for, they're probably still not accessible through in vivo or in vitro experiments. So this is why this whole project started, indeed, because there were a few things that were more practical to investigate using in silico models. So thinking about your very comprehensive model of the activity of a neuron and astrocyte and the perisynaptic astrocyte, what's your take on metabolism and calcium?
Yeah, I must confess that I'm a beginner in thinking this calcium and metabolism. I remember when I started this project that I presented today some 8 or 10 years ago, I was reading quite carefully about Pierre's papers and trying to understand this lactate and other issues like the metabolic aspect of it. But then I decided it's too complex for me, and I will concentrate on the calcium story only. I would first like to point out that when we do when we do modeling of this calcium in astrocytes and also in neurons, so we could also get a little bit ideas, like if we look at analogies from neurons. So first of all, there we have to see like that calcium is very compartmentalized in neurons, and like all of us know, and it's linked to like different physiological phenomena depending like where you look at the calcium, whether it's dendrites or soma or and which aspects, and most likely it's the same in glial cells, I would think. Then the second aspect is that like in terms of modeling, which I am more like where I have more expertise, is that I should point out that the astrocyte models that we are right now using, they are in a very beginning. So if you compare, for example, how you model the ion channels, the receptors, pumps, and so on in neurons, it's much more developed. In astrocytes, we are actually loaning a lot of data and ways how we model these entities from oocytes and also neurons. So it's kind of like that we are still lacking data, but this is we have to start somewhere. And like we always, like in our case, we report where we have taken the data and the model from. So that is one limitation. And I like myself, like there were these suggestions like the sodium pump and other things. So what I consider is lacking in the astrocyte modeling is the modeling of the astrocyte cell membrane as such. So there are no good models for that, and definitely clearly I see that I should also make some improvements there, since I've been doing astrocyte electrophysiology myself earlier. One of the reasons why this has not improved is that actually for example, if you would like to incorporate voltage-dependent channels in astrocytes, it seems that they don't work, they don't obey Hodgkin-Huxley kind of kinetics. So there is a need for new types of models, even for kind of like for voltage-dependent channels, and of course this kind of influences like how you introduce the regulatory environment for like ions and pumps and so on. So we are really lacking in the glioscience modeling the proper models of the astrocyte cell membrane, and hopefully I can contribute to in some aspects to that as well in the future. The question was related to calcium and metabolism. So I sort of got an idea, like in the beginning of these talks, like I think it was Pierre's talk, like when you again explained this lactate story, and I understood that during development, maybe also later on, you need this BDNF and other stuff to sort of kind of prepare the synapses to do like what they are supposed to do. This is how I concluded it myself. So I'm kind of thinking myself that these things, of course, they can be interlinked, this calcium and metabolism, but it could be also that they are kind of like separate physiological events that needs to be like for the proper functioning of the brain and proper learning and proper like let's say enhanced memory. They need to occur in certain order, and like now when we are modeling, we are just modeling one aspect of it. Like in our case, maybe the modulation of lactate needs to occur before these kind of like subsequent modulation by different neuromodulators may occur through astrocytes. And it's a very delicate system, and we cannot kind of like it's kind of like that they all exist there, but depending on the circumstances and the need, they are recruited, and especially during development, they have to happen in a certain order in order not to run into a neuronal circuitry that is not able to process the sensory information the way the rest of the rodents or humans do. So you need to have these things in place, and they may occur in a sequential manner. And like in specific synapse, like the one we now model in rodents, of course not in humans, but rodents, and I suspect that the events that I'm modeling are related to actually pruning that synapse. Like you need to prune synapse also, like when you develop, so you cannot go on with all the synapses. So maybe there are like many physiological events and many signaling molecules that kind of drive these so that you mature synapses, make proper networks, gradually start developing the plasticity and learning, and then finally secure the memory. So you need to introduce like different signaling molecules. And maybe like I'm not a biologist originally, so maybe this is too strong to say, but maybe we are kind of like, we consider there is something signaling when we've seen that it's related more to kind of like synaptic transmission or sensory information processing, and then other molecules we consider they are like metabolic molecules because they may more originate from the vascular system. But what if we should think about this, like some of you are already hinting that it's all signaling, and we shouldn't kind of like categorize things in such a way. I myself think that this neuronal astrocyte vascular entity, it's kind of a one information processing unit, and all those aspects that we see, whether they are fast or slow, they are needed. Because if you think like from the theoretical or engineering perspective, a dynamical system, both fast and slow processes can coexist, and they may have a meaning for the system. And this, I think this was my message also with our model that I'm trying to convey, but it's maybe not so evident yet what I'm trying to show with the model we developed.
No, I find your remark interesting. Just anecdotal. I think none of you were active in the late 70s, but I was just starting. And glutamate was not considered a signaling molecule in the brain because people were thinking it's a part of the intermediary metabolism. How can it be specific? Well, now we know it's quite important as a signal. So, and there are now examples from other areas. I mean, lactate, of course, but not only in the brain. I mean, in immunology, it's changing the properties of T cells. And one can speculate. I mean, this is a pure speculation that early in evolution, there weren't that many molecules to kind of in multicellular organism to essentially the number one concern was energy. Make sure you can keep the electrochemical gradients and your internal milieu, and that costed energy. So you used whatever you had, of course, in terms of energy production, so intermediary metabolism. But then if you wanted to be a bit smarter and send some information between cell types, you use the same molecules, and eventually only later it became more sophisticated. I mean, just an idea.
Thanks everyone for your remarks. Indeed, one of the things that I would like to point out, indeed, is one common thread in the whatever it's been taught so far by Felipe, by Pierre, and by Marja-Leena probably. So the window of observation that we're taking, which is somehow mediating the effect from an entire cell probably, or from a certain subdomain, like Marja-Leena, who's really trying to model whatever is happening at the level of synapse or right around the synapse. So And I think this is one of the questions that our KAUST BBP consortium has been trying to tackle. So how can we turn these point models that we do into really accurate geometric model, and how these could change our view on the effects that we observe, which leads me to a question that I would like to pose to Johanna, because she's the one who is expert indeed in visualization, and she has shown some a lot of very interesting approaches that somehow I think goes in an opposite direction to what we do. So we're trying to be very specific about what happens and quantifying in a very specific way of the mechanisms that happens around individual synapses and individual astrocytes, and then all over the cell. But indeed, what Johanna and her team did was to try to simplify this, right? Like the work that you did on neurolines, where you consider the neuron as and the dendrites as a station with all the spines, which I think is a quite clever way to interpret the data from individual cells. So my question was whether and we've been working together on this to some extent, whether now that you know astrocytes a little better, whether you think that this kind of approach can be somehow extended to astrocytes and help our research towards metabolism and astrocytes to shed light on some of these very complex mechanisms by taking a complete opposite direction. So we have this tendency of complicating a lot because we want to understand everything, but on the other way, somehow we should try to simplify things and to abstract the problem.
Yeah, absolutely. I think that would be a super interesting research direction, right? I mean, what we did for neurons is that we kind of tried to, like you said, in neurolines, we tried to simplify like the 3D structure of neurons and their connections into like an abstract 2D subway map where every neuron was represented basically like a subway line, and we kind of straightened the edges, and it was a 2D representation instead of 3D, so that it doesn't give you like occlusions and difficulties in interpreting it. I think that exact approach is still a little difficult to apply to astrocytes because their structure is just so complicated, right? With neurons, you can think of you're straightening them out, flattening them, and that will kind of work. Astrocytes so far still seem too complex in their structure to use the same approach, right? But it doesn't have to be the exact same approach, right? It would be just thinking about what are the abstractions in astrocytes you guys are interested in that are important to keep, and what can we kind of remove, what is not as important, right? So it's often like a lot of dialogue and figuring out what can we simplify and what are the tasks you're super interested in that we need to support to come up with like a visual encoding that then simplifies a lot of what is currently distracting and difficult to kind of quantify your analysis.
I see a hand by Felipe. So
oh yeah, this is thanks. This is
points. So please.
This is a question for Johanna. You know what? We love mice. We love rodents and Drosophila and all that. But finally, we're always thinking about humans, right? So my question is, have you had any chance to model human tissue? I know the EM is probably very difficult. Tissue is not very good in postmortem samples, but for instance, it's been proposed that astrocytes in humans they do things differently than in rodents. In terms, it's not just they are that bigger, but they might do things which relate to what is different between primates and rodents. So have you had the chance to walk into these human astrocytes and compare to rodent astrocytes?
Right. So I personally have not, but I know that in our research group, we do start to have like a data set of a human brain. So we could do that in the future, which would be very interesting to do, to kind of, that would extend like the comparative aspect as I talked about before, right? Where we might not want to just look at different neighborhoods in one data set, but maybe compare different data sets. And that could be like both of mice, but at different developmental stages, or it could be between a mouse and a human.
And also Drosophila, you know, for some reason that nobody knows, in Drosophila, most of brain synapses are not glutamatergic, are cholinergic. And would that evolutionary decision that happened many hundreds of million years ago, would that have a structural impact? Maybe you, with your visualization system, you would see and they say, oh, these are different in maybe relations between glycogen granules and synapses and whatever. That would be very exciting to do that comparative biology.
Absolutely, that sounds really fascinating. Yeah, I agree.
So I would like to take the chance to be a bit provocative with the panel and maybe try to shoot you a bomb about something I haven't prepared you. So Johanna was mentioning relating to what we have been working about, what we have been working on, that we did this exercise many times actually. This is really interesting. So compared to the kind of analysis that we want to do and the kind of process we want to investigate, which kind of abstraction would be more interesting to do at the level of the cells, at the ultrastructural level of the cell. So I know that it's a kind of a very vague and general question, but if you should think about something astrocyte is doing and some process you're investigating, which kind of ultrastructural correlate to the process that you're looking to, you would take as a potential candidate to try to disentangle, let's say, the complication of the structure of the astrocyte and try to look into it, like ER, mitochondria, or stuff like that. A three-dimensional organization between the astrocyte and its environment that would give you probably a different view on its way of interacting with everything that is outside its territory. Was I vague enough?
Yes.
Well, if I may start.
Yeah, go.
You know, when you think about local domains of metabolites or calcium and protons, the one that is really very little studied is NADH. So Pierre was mentioning regulation of NMDA receptors by NADH. And NADH can be very local. This is interesting from the structural point of view. I wouldn't expect a lactate gradient between a process and the soma in astrocytes. We have done the modeling and that is very unlikely because as Marja-Leena would say, these are well mixed compartments in terms of something that is millimolar. But NADH being nanomolar and protons and calcium, those are the three things that you really need to know where things are in relation to each other. So I would love to know the spatial relationship between NMDA receptors in astrocytes, other NADH sensitive proteins, which are channels in general, mitochondria, and the glycogen granules. I mean, how those three things are structurally related. Because if you have a local NADH domain produced by, say, lactate dehydrogenase or GAPDH, maybe that will be very relevant. For instance, hexokinase doesn't matter where it is in the astrocyte because it's a well mixed, and we don't know why it's attached to mitochondria. This is really mysterious because it's nothing to do with the metabolism of glucose. This is some regulatory function that we don't know. So eventually, when you have EM data in which you can stain things very precisely, which I guess is not there yet, that structure would really be exciting for Johanna to really understand. That would be my future Christmas wish.
So somehow takes us to mitochondria, I guess.
Yeah.
Pierre, do you want to add something? I hear you very silent.
No, I mean, I think it's mitochondria in astrocytes, and I'm not saying this just to be nice with Linne, Marja-Leena, but I think, they, because of this approximation, which is correct, that astrocytes are mainly glycolytic. So, okay, well, the idea was even they have very few, I mean, again, sorry to always go back to old history, but, I'm a bit more mature than you guys. When I presented this, you say, one of the guys said, but I guess Pierre forgets that there are mitochondria in astrocytes. No, I didn't forget. So just to tell you. So, but why, for example, I mean, there are many explanations, by the way. pH, the Crabtree effect, the phosphorylation of the PDH. I mean, there are many. And then today we heard about how potassium can decrease respiration. So there are many reasons why it's predominantly globally glycolytic, but probably things are much more. It means, I mean, if there are so many mechanisms, I mean, Bolanos showed NO also plays a role there. So there are many regulators of mitochondrial activity in astrocytes. It means that it must be a very important function, which then, of course, where these mitochondria are localized, maybe they're differentially regulated depending on where they are. I don't know, but it's definitely an obvious intracellular organelle, very important in coupling activity to metabolism and signaling, probably.
I'm interested to hear because with Johanna we are trying we're trying to design this tool where we try to look whether mitochondria is more related with dendritic shaft or with spines or with axons or with boutons. Where would you think metabolically its presence would be more relevant?
If I may say, I mean, it really depends what the mechanisms would be involved in the regulation of the function of mitochondria. In a way, okay, location is important, I agree, but maybe location depending on what signals will act on these mitochondria is even not more important, but you need also the signaling to understand the meaning of the location, I think. But places where again, let's take the example of NO, for example, or potassium. I mean, the signaling through, that's pH then, because it's the NC that would be involved. So protons, and then also we showed also the role on of pH. So it's important to know where they are, I agree, but maybe it's also important what kind of mechanisms can be active where on mitochondria.
Marja-Leena, from your modeling perspective, I mean, does your model integrate the activity of mitochondria? Or I mean, that's pretty metabolic, right? So probably you haven't modeled that in detail, or little or none. So what would you? How do you think it would change? Knowing that it's still a point model, so probably there's little effect on from a morphological and geometric point of view.
Yeah, we don't directly model the mitochondria. Of course, we could sort of think like that we implicitly take it into account when we are modeling for example, the presynaptic terminal and the astrocyte terminal, but we don't really precisely model it. But I've been following a bit like for the past year or so, like the calcium signaling, and when these signals have been measured from different compartments of astrocytes, both in vitro and in vivo, and to me it looks like that in some, now I don't remember the details of the very recent studies, but there I understood that in some processes, in some developmental phase at least, and in a kind of spontaneous situation, the mitochondrial calcium was really like the driving calcium force in some compartments of the astrocyte. So clearly the mitochondria in astrocytes should be also modeled. I agree that, but now it's mostly the ER that we have concentrated on, mainly for historical reasons, because many of these models, they typically model this ER. If I could comment on the like from the morphological perspective, like what would be required for the future modeling. So when I go back and try to think about the model construction process, so for us, one important thing from the biological perspective was a bit lacking, like the kind of understanding, like in general related to these mechanisms that we model, that very rarely it's kind of like assumed that it's enough that you present like at this developmental phase, this mechanism was there, and we saw these influences. But for me as a modeler, it would be really important to kind of like have a better understanding, like if you look at, for example, the different postnatal developmental phases, that I would get information like which mechanism exists when. This would be really crucial, because now it was a bit guessing, and I needed to dig out, and I actually needed to leave a lot of references also out from our model, because they were not really comparable to the developmental phase that we were looking at. And this is what I have learned during this modeling process, that many of these mechanisms, at least some, seem to be really developmentally regulated. And I was also kind of like myself thinking that whether this is this has some, of course it's kind of like it has developed through the evolution and it has some meaning, but then on the other hand, like is this a way, again going back to my earlier discussion, that is this a way like how you recruit these different metabolic and signaling molecules that exist there, that your target mechanism keeps changing during development, and then later in adolescence and adulthood, and all those signaling and metabolic molecules exist there, and they may be used in some signaling pathways, but depending on the target mechanism, and this may change with time. So you sort of introduce the new behavior or physiological phenomena. So for me, it would be very important, like if you can see from your EM reconstructions, like which mechanism exists when, to have a better idea about that. So that would be really nice for the modeling.
Yeah, I think for that you would really need to combine a little bit of or quantitative immunohistochemistry, which which is
Yeah.
feasible, but there's still not much because I mean the way you produce zero sections to a certain extent combined with 3D EM has some limits, which are classic problems related to the penetration of the antibody. But if you're limiting your observation to individual synapses, probably that could be an idea. And I mean, it's something on the pipeline, but you know, we are.
I know it takes time.
Too many things to do and too many things we want to do, and we have to give priorities. But most certainly, setting up the technique would be most certainly awesome. To conclude, there is one question from the audience actually, and I think it would be a good way to conclude this session, because it can give us a way also to discuss pathological models. So they ask from the audience, what are your insights into the role of cytokines in mediating the astrocytes neurons crosstalk? Cytokines could interact with the astrocytes at the BBB without needing to penetrate the neural tissue at all. So I don't know, anyone, Pierre wants to?
Yeah, I mean, I can say that definitely cytokines, in pro-inflammatory cytokines, IL-1 beta and TNF alpha, drastically modified the metabolic phenotype of astrocytes. Fundamentally, astrocytes become more, as I used to say, egoistic. Their metabolism own metabolism is activated very much. They use more glucose, they use all the glycogen they have, they decrease lactate release. So they obviously, there is actually, it's a large field of investigation. If what one was a thesis of an MD PhD student. She did a couple of nice papers, and then there was a postdoc. So we have 3-4 papers on this topic, but that was even more than 10 years ago. And it's certainly out there open to be, you know, particularly now with the omics that are available, it would really be interesting to look at this metabolic plasticity following pro-inflammatory cytokines. So now, whether these are cytokines coming from the circulation or formed within the brain, that's okay, that would be interesting, but definitely astrocytes are targets for cytokines, and their metabolism is really quite radically modified by cytokines.
Do you think this could be maybe a feedback mechanism from the body that could trigger, in a way, neuropsychiatric conditions? And so astrocytes could be a gate to enhance troubles, the psychiatric troubles in patients.
Well, again, I'm biased, but I really think that, and I'm not the only one, think that astrocytes dysfunction is involved in mood disorders, in depression. I mean, there are evidence from brain banks looking at expression of genes, and you know, Huda Akil, for example, she kind of runs a huge biobank in the US, many collaborative centers, and she mentioned more than once that actually the genes that seems to change most are astrocyte-based genes. Plus, there are many published papers showing a decrease in the density of glia in general, but astrocytes also in subgenual prefrontal cortex, in I think dorsolateral prefrontal also, all areas involved in major depression. And then, as I showed you, we found that lactate, which is essentially produced by astrocytes, has an antidepressant effect. We're trying to understand the mechanism.
Felipe, you want to maybe give your insight on the role of cytokines coming from BBB on astrocytes and maybe a possible metabolic?
Yeah, I would make it more general. So bloodborne substances that affect astrocytes. This is something that we haven't really tested very much. we are focusing on the internal regulation. But for instance, during exercise, there are few substances which are released, like myokines, that have been proposed to affect astrocytes and neurons, and astrocytes regulating the access of those myokines to neurons. And then the big keyword there is the blood-brain barrier. For instance, psychosis are induced by just small changes in blood-brain barrier permeability, because glutamate, which is in the blood at 200 micromolar, goes in. Imagine 200 micromolar for an astrocyte or a neuron is a major problem. And then other substances also go in, like cytokines, which are circulating, or myokines, or others. So this is a world that we don't really know very much about it. And lately, there's been 2 or 3 papers on short chain fatty acids produced by the microbiota, which can travel through the circulation and affect mitochondrial metabolism in astrocytes. This is something that it was touched about 20 years ago by people looking into this question of which is the major substrate for brain metabolism that we always assume is glucose. It is true that glucose is the most important, but these minor substrates like fatty acids, which are apparently metabolized only in astrocytes, are something we don't have numbers about. The research has been qualitative. So we know the enzymes are there, the factors are there, mitochondria in culture astrocytes can respirate very easily with the fatty acids. But then in the brain, we don't know. Is this 5%, 10%? Is it that normally astrocytes are using oxygen from fatty acids and maybe not at all from pyruvate, and all the lactate is going away? And then these are really deep biochemical metabolic questions that we are not addressing. There are the guys that were interested in this kind of questions are retiring, and this is a big hole in the field, because metabolism is not solved. It's really starting, and this is very interesting maybe for the young people in the audience that with new tools, with new techniques, they would like to tackle those questions, because it's much more complicated than we think, and much more exciting. Going back to cytokines, if you add cytokines to, we have done that in cultures, everything changes in an astrocyte. They're really sensitive to the cytokines. So it's a different cell, as Pierre said, the selfish astrocyte. And how many of those are in a brain or in a peculiar area of the brain that might have to do with the neurodegeneration? You know, these are important questions. So I think the person interested in cytokines, they should do the experiments.
Absolutely.
Well, I published a paper on effect of the CXCL12 on CXCR4, which is a receptor, GPCR receptor expressed on astrocytes, and this was done in culture. But basically, we were activating astrocytes with CXCL12, and then the astrocyte was responding by releasing glutamate. So this was supposedly a mechanism feedbacking from cytotoxicity, where with a certain level of inflammation, then you would increase the usual calcium, intracellular calcium, and then trigger release of glutamate. So I think, as you said, it's not a new story, most certainly, but from a metabolic point of view, I was unaware, let's say, or it's very interesting, the fact that exposing astrocytes to cytokines changes the metabolic phenotype of the cell, which would, of course, being detrimental from anything that is around the astrocyte, right? I don't know, Marja-Leena, you want to add something about cytokines on astrocytes? You have your personal take, or?
I'm not an expert either on this. I'm sorry. I've been studying the previous work by, I think it's Volterra and some others. So that's the only thing I know. But maybe I would like to more comment on a general level, like about the role of astrocytes in pathologies or let's say neurodevelopmental disorders that I have been looking more recently lately. For example, schizophrenia. And Pierre already mentioned that in mood disorders, there are like changes in genes, and the same we see in schizophrenia. We see changes in genes that encode calcium related aspects in neurons, but we see a few changes also in astrocytes, and very interestingly, but these are all unpublished results, and well, I don't have any final conclusions so far, but when we take the human pluripotent cells from control groups, and then these very severe cases of schizophrenia groups that are kind of like diagnosed by the medical doctors. So we see really crucial differences in cell cultures that we when we look at the synaptic transmission, for example, and the morphology, first of all, synaptic transmission, the dynamical behavior of neural networks. But of course, it's again like a very controversial setting, in the sense that you take human cells and you do a lot of manipulations before you culture them. But at least we see like between different control groups and between different schizophrenia groups, we see big differences in the behavior of astrocytes and how they influence the two-dimensional networks. So it's not three-dimensional, but two-dimensional. So surely there is something. It's not related to cytokines probably, but something else. But they have a big role in my mind in neurodevelopmental disorders and diseases.
Yeah, if I may say, I mean, I think again, there is a little bit of a hype and also controversy, but certainly using patient cells and iPSC-derived astrocytes or neurons, it's really a worthwhile avenue for hopefully trying to close a little bit the gap between basic neuroscience using rodents, which are fine, we learned a lot, and maybe diseases in the brain diseases, including psychiatric diseases. I mean, we have a collaboration with Pierre Magistretti at University of Laval in Quebec, and he is producing iPSC derived neurons from patients, psychiatric patients, and he sees definitely some differences. I mean, it will be hopefully submitted soon, and so again, we maybe just adding some more noise, but maybe not. Maybe there is some important mechanism that can be revealed using these human cells from patients.
Right. I think it's 10 to 6, so our session is probably over. I think we had a good panel discussions, so I want to thank each one of you for your interventions and your very interesting insights regarding this very big topic about the future of glia research, and hoping that you send some hints to our younger students and our younger scientists in the audience, because there's still very much to learn and a lot of experiments to do. So thank you very much. And I think if no one else has anything to add, I could adjourn our workshop to tomorrow afternoon.